So each year, the Shared Knowledge Conference is a celebration of UNM graduate students and their outstanding research and scholarship. The conference provides a venue for students to engage with UNM and larger New Mexican communities, to cross the borders that too often divide academia from the larger world. And in doing so, we spark conversations and work together for a world of equity, innovation, discovery, and growth. Thanks to all the students that contributed posters and prepared LoboBites. Some of these posters are going to represent UNM at Graduate Education Day at the state legislature this February. Additionally, our LoboBite finalists will represent UNM in Tucson at a regional competition, so we look forward to that. I need one more hand. UNM's LoboBites is a version of our 3-minute thesis. So a LoboBite is a soundbite distilling researcher scholarship into a short, pungent, and perhaps catchy monologue. So for the most part, we adhere to the trademarked 3MT rules. That's so that we're eligible for this regional competition. So those rules include that students are allowed to have one static slide. They get a 3-minute timer that'll be on this screen. It'll make you as nervous as it makes them as you see it count down. And they have a judging panel. And because they're supposed to speak to a broad audience, our judging panel are non-academics and fairly distinguished. And I look forward to introducing you to our judges at the end of their LoboBites while we're tallying scores. So you'll meet them then. But they will be judging equally on significance, clarity, delivery, and engagement. These students participating were all in preliminary heats last week, and they were chosen from those preliminary heats to participate today. I want to remind you that there is a chance for a People's Choice Award. So when you walked in and sat down, there was a program on your chair. You use that program to vote for who you thought was the best, and at the end of the day, we'll be collecting those, and that person will get an award as well. So our first speaker today is Phuong Nguyen. She'll be speaking about improving the palatability of colonoscopy preparations.
So we all know that cancer is really bad, but what we really often hear of is like breast cancer, lung cancer, prostate cancer, and etc. What you don't really know is the second leading cause of cancer-related deaths in the United States is colorectal cancer, and 100,000 new cases are diagnosed each year in the United States. Globally, it's expected to grow by 60% by 2030, so 2.2 million cases annually and 1.1 million deaths. And interestingly, this is something that's highly preventable and highly screenable by a process called a colonoscopy. So a colonoscopy is something where they visualize your stomach and then they remove precancerous growths or polyps from your stomach lining and prevent this from ever happening. But only about 50% of the population actually go and do what they're recommended to do. And the reason that many people say is because the prep is disgusting. And for those of you who don't know, it's on the left, and imagine you're going and you get given a jug, and it's 1 gallon. You get a packet, it tastes kind of like lemon, and it has salt and the drug, and then you're supposed to mix it with 1 gallon of water. And then every 15 minutes, you're supposed to have an 8-ounce glass, and it kind of is slimy and salty and lemony, and then you drink it, and you kind of want to gag, and then you vomit, and then you come back, and then you keep doing it so that you're actually through and you actually finish your colonoscopy. Well, we're working on developing an alternative so you don't have to go through this process. We thought, what if we encapsulated it, kind of like boba tea pearls on the left, and we called it Bubblite. And if you encapsulate it and it's stable, so you can drink it in anything you want, smoothies, and if you know the exact amount of drugs, we can start testing and say somebody like me doesn't get 200 grams of the drug, and they can get something a little bit smaller, so you get less, discomfort and nausea and vomiting. And, and you also get to kind of not have to taste that flavor. So I think we can actually reduce colorectal cancer rates and colonoscopy preparation so more people would do it and reduce those rates with something as small as tapioca pearls or bubble tea. Thank you. Oh gosh.
So PHUONG, tell us where you're from.
I grew up here, but I was born in Vietnam.
Okay. And what do you plan on? Do you plan on doing something with this?
So actually, this was something we got into the Lobo Rainforest NSF I-Course site program for with my advisor, and we kind of wanted to move forward and start testing it on mammal animals because it's already biocompatible.
Okay. When you test it on animals, part of it is that you don't like the way it tastes. So how do you find that out from animals? If it's an improvement.
But it's an improvement. So one of the things is we have to prove that it's something that you can eat and drink without killing the animal. And then you can actually migrate it into, I know this sounds really terrible. And then you can migrate it into people and actually taste it. There's ways around getting approval if you're willing to test it on yourself, which my advisor is willing to try on premises.
Okay, thank you so much, PHUONG. Our next speaker is Suzanne STRADLING, who will be speaking about hydroelectric management on the Rio Chama, balancing competing ecological priorities.
So, my name is Suzanne STRADLING. I'm an environmental and energy economist, and I'm here to talk about saving the world. So, I stumbled into this saving the world gig. I actually just meant to look at the economic impacts of changing water flow at the El Vado Dam on the Rio Chama. So, I looked at the power implications and the reservoir recreation implications and the tourism implications, and then I looked at the environmental values associated with it, and that's where it got really interesting. The Rio Chama is beautiful. It's a wild and scenic river, and it's one of the most biodiverse areas in New Mexico. I think we all understand that there is intrinsic value to that. But there is another environmental issue at play here. Electricity production in the United States is responsible for over 1/4 of greenhouse gas emissions. If we want to avoid climate change, we need to do something about that. Solar panels are our golden ticket to a carbon-free grid. But the problem with solar is that when the sun goes down, it stops producing energy. At the same time, it gets dark and we all flip on our lights. And so what we see is a dramatic increase in demand for electricity. And this increase is more dramatic the more solar we have on the grid. So what do? Batteries are not sufficient. In 2020, in California alone, we're talking about 13,000 megawatts of energy that has to come online in a little less than 3 hours. That's enough energy to power 13 million homes. What we use instead are natural gas or jet turbines. These are basically jet engines. They're dirty and they're expensive. We could choose to increase our hydroelectric production from the 5 to 8 o'clock hours. That would decrease our emissions, and that would increase our ability to use solar panels that exist on the grid and to introduce new solar panels onto our system. Why don't we? Well, it has real implications for the riverine environment. It degrades beaches. It injures species. This green versus green trade-off is at the core of my research. So how do we value the environment? We ask people. We want to know how much New Mexicans value this particular stretch of the Rio Chama versus their value that they put on decreasing greenhouse gas emissions by a certain amount. When we have that answer, we can better manage the Rio Chama, but we can also start looking at other dams. This is important because hydroelectric dams store an immense amount of energy. They are the big batteries that we need. And they may be our ticket to a zero carbon grid, and yes, to saving the world. But we have to know our trade-offs. We have to know the pros and the cons. And my research gives us insight into the questions we need to ask and the tools we need to use so that we don't destroy our environment as we try to save it. Thank you.
Suzanne, this is your opportunity to tell us why economists will save the world.
The fact is, we all have great ideas, but we really have to choose the ideas that make the biggest difference. We have to go for the big impact thing, and that's one of the things that the cost-benefit analysis that economists do can give us. I think that's really important. I know it's not sexy and fun, but it's really important.
And where are you from, Suzanne?
I'm actually from Los Alamos.
And what do you hope to do with this when you're done?
I'm really interested in the ways that we're going to change the electric grid over the next 20 years. And so I want to be researching in that area, seeing what happens to our capital investment in electricity, particularly as we look at our issues with carbon production.
Great. Thank you so much.
Thank you.
Our next presenter is AYUSH SHAHI, speaking on Is Wastewater Useless?
So what if I told you you could take stuff out of wastewater and turn it into something useful? Is wastewater completely useless? Perhaps not. We're working on a project where we're trying to treat the wastewater coming out of coal-fired power plants in a way that it can be recycled back into the system. And we're also trying to generate commodities along the way. Good afternoon, everyone. My name is AYUSH, and I'm here to talk about my research on wastewater treatment and recovery. In my opinion, the biggest challenges of our times are the management of energy and water resources. Could you imagine how our life would be without access to electricity and water 24/7? I couldn't. Coal is not the most green source of energy, but there are still many existing coal power plants across the United States. Currently, 39% of electricity is generated from coal. There are many infrastructure present, and there are ways in which we can make them more environmentally friendly. And we are proposing a treatment processes where we're incorporating various physical and chemical processes such as ion exchange, nanofiltration, membrane distillation, and precipitation to get the water of a certain quality which can be recycled back into the system. Because typically coal power plants draw in huge amount of water for their cooling towers, and if we can recycle the water back in, that will decrease the water demand of the power plant overall. This becomes a relevant topic in water-stressed areas such as New Mexico, Arizona, and Texas. And during our treatment process, we're trying to extract commodities such as gypsum and brucite. Gypsum, as many of you might know, is used to make walls, boards, and ceilings, and brucite is used as a chemical disinfectant. So this gives an economic incentive for power plant owners to opt for our treatment process. And we hope that this process will be a win-win situation for power plants as well as agencies trying to maintain environmental regulations. Thank you all. I'm AYUSH, and my talk was on generating money from something as trivial as wastewater. Thank you.
Thanks, AYUSH. Tell us where you're from.
I'm from Nepal.
And what brought you to New Mexico?
So when I was in Nepal, I heard about UNM for Nepal, which is led by one of our faculty members, Dr. Mark Stone. And then I got to know about, for those of you who don't know, it was a post-earthquake a project led by students and faculty here after the earthquake hit Nepal. And that's when I heard about it and decided to apply here.
I didn't know that, and actually last year one of our LoboBites was on the program UNM for Nepal, so that's very cool.
Oh, good.
Great. Thank you so much, AYUSH.
Thank you.
Our next presenter is Lauren BANSBACH, who will be speaking on ponderosa pine seedling physiology after severe wildfire. Does it take a forest to make a forest?
Hi, everyone. My research is about ponderosa pines and fire. Many of you have probably heard about all the wildfires across western North America. For ponderosa pine forests here in the Southwest, small, low-severity wildfires leave most of the trees alive and are a natural part of this forest ecosystem. In contrast, big, catastrophic, high-severity wildfires kill most of the trees and result in huge areas of the landscape that are no longer forested even decades after the fire. And even efforts to replant trees in these severely burned areas are often unsuccessful because most of the seedlings don't survive. And so why aren't our forests coming back after severe fire? Well, one obvious difference is that in severely burned areas, there are no more trees. And so it leads me to the question, does it take a forest to make a forest? In other words, does the presence of large mature trees overhead have some sort of beneficial impact on the small developing seedlings down below? To test this, I'm studying ponderosa pine seedlings growing in these two contrasting habitats, a low severity burned area with trees present and a high severity burned area with trees absent. And so one reason I expect a difference between these two habitats is because if you've ever gone walking in the woods, you probably noticed that the presence of big trees overhead actually changes the amount of light, heat, and moisture that you're experiencing. And that's called microclimate. And without trees, the local microclimate is hotter, brighter, and drier. And small seedlings are especially sensitive to changes in microclimate. They can experience great physiological stress under dry conditions, and that's made even worse when in combination with high heat and excess light. And so to relate to these seedlings, just imagine being dehydrated, standing out in the hot, blazing sun all summer long. And so to quantify differences in these seedlings, I'm measuring several plant physiology metrics that relate to light, heat, and water stress in these seedlings. And interestingly, my preliminary results show that the seedlings growing in the severely burned area are significantly more stressed than seedlings growing in the low severity area. So this suggests that maybe it does take a forest to make a forest. Ultimately, the results of my research will help us understand why our forests aren't coming back after severe fire, and they'll help land managers seeking to successfully replant trees after severe fire, and it'll help restore our ponderosa pine forests. Thank you.
So Lauren, tell us where you're from.
So I'm from Missouri.
And what brought you to New Mexico?
I came here for the biology graduate program.
And anything in particular attracted you to the biology, a particular advisor or?
Yeah, so my advisor is great, and I also wanted to study plants here in the beautiful desert Southwest.
You can give a shout out to that advisor.
Oh yeah, David Hansen, and Matthew Hurteau, also Marcy Litvack.
And is Albuquerque what you expected?
Yes, it is much better than what I expected. I didn't really know what to expect. So.
Do you have a favorite forest here?
Oh, well, they're all great.
Okay.
But,
Thank you. Our next speaker is Ty Martin, who will be speaking on computational investigations of the intersections between bioactive compounds and biological assemblies. Ty.
So today I'll be talking to you about Alzheimer's disease and the critical need to develop sensors for early diagnosis. So Alzheimer's actually is the 6th leading cause of death in the US currently, and it affects about 6 million people right now. But what if I told you that number is going to reach 14 million by 2050? So right now there's really no way to diagnose Alzheimer's in an early state. In fact, the only way to really do so is after death, when you actually observe the brain and see the problems that arise. And so Alzheimer's has 2 specific features that you see in the brain. They are called amyloid plaques and neurofibrillary tangles. And these are basically large lesions that sort of disrupt the communication of your brain. So if you imagine the brain being something like a power station with the lines reaching the rest of the body, well, these plaques sort of disrupt that communication, and so your brain no longer functions properly. And the plaques specifically are made of these proteins. And these proteins are actually naturally in the brain, and they have a function. But in Alzheimer's, they misfold and clump together or aggregate to form these plaques. And so this brings me to the critical research that I'm involved with, which is to develop a way to diagnose this early on so we can begin treatment sooner. And currently there are a class of specialized, unique small molecules that are developed by the Witten group here at UNM, and I'm actually involved with that group as well. And so my research specifically is using computer simulations to understand how these special molecules can be used as a sensor. And what happens is these molecules, we call them OPEs, they're a little bit like a light bulb. So they bind to these protein aggregates and give off a measurable signal. So it's like the light bulb turning on. And the reason that we think this happens based on my simulations and our experimental results is that the OPEs bind to specific places on these aggregates. And when this happens, there are something called intermolecular forces going on. So if you imagine magnets sticking together, it's something similar to that. And so what we hope is that we can engineer and further refine these OPEs to be able to be used for other types of neurodegenerative disease, like maybe Parkinson's in the future. And so we hope that eventually our research will lead to early detection and hopefully a treatment and cure for this disease that affects so many. Thank you.
So Ty, when you talk about the future, how far into the future are we talking about before some of this has some application?
Right, so right now we're actually testing the OPEs on mouse and rat models that have Alzheimer's. And so we hope that, I'm thinking maybe in the next 10 years we could try some clinical trials. So
I think I'll probably need one, so I'm ready. Where are you from, Ty?
I'm from here, from the Moriarty and East Mountain area.
Okay, great.
Yeah.
And what made you pursue this line of research?
Right, so I actually have a muscular dystrophy which has proteins that misbehave. And so that sort of piqued my interest, you know, finding ways to sort of help treat these diseases that are serious. So
great. Yeah. Thank you so much, Ty.
Thank you.
Our next speaker is Julie Allison Spencer. How does antibiotic resistance spread in tuberculosis?
My grandma had an antibiotic-resistant infection for the last 11 years of her life. That's why I chose to focus my dissertation on antibiotic resistance. So zooming into a specific disease, tuberculosis is alive and well on the planet. Over 10 million people every year become infected, and 600,000 of those cases are antibiotic resistant. Yet the treatment effectiveness rate for those is only 54%. This situation could become worse. Tuberculosis is caused by a bacterium, Mycobacterium tuberculosis, which usually multiplies in the lungs. It passes through the air by coughing, and after a resistant strain has evolved in a person's lungs, that also passes through the air exactly the same way. But is this mechanism sufficient to explain the incredible diversity of TB strains we see in the world today? Resistance can spread another way, by horizontal gene transfer. Here's an illustration. I have blue eyes. My daughter inherited blue eyes from me, but what if I could give the genes for blue eyes to my friend? That would be horizontal gene transfer, and it's what happens every day when the genes for antibiotic resistance jump between cells of bacterial pathogens. Most researchers do not believe this is happening with TB. However, there are 3 different kinds of horizontal gene transfer. The first is when a gene swims through a tube to get into the other cell. The second is when it's sucked in from the surrounding liquid. And a third kind is when it hitches a ride on a virus that attacks bacteria called a phage. Could phages be spreading the genes for antibiotic resistance? I investigated this question by analyzing 67 whole genomes of antibiotic-resistant tuberculosis, and I was looking for regions in the DNA that unexpectedly showed sequences recombining. I found 33 signatures of recombination, and some of those had antibiotic resistance functions. So we already knew that resistant TB strains pass through the air, but now we suspect that phage could be spreading the actual genes for resistance. I hope that my research leads to new treatments to help alleviate the kind of suffering my grandma had to go through.
Julie. Tell us where you're from.
I'm from Los Lunas, New Mexico.
Great place. I have some roots there, as we talked about as well. So you got interested in this because of your grandmother or for other reasons as well?
Yeah, so when I was a teenager and my grandmother was in a nursing home with this infection that just would not go away no matter what medicine they gave, I had that question of why isn't the medicine working? But then when I went to graduate school in biology, I became interested specifically in bacteria because they have a short generation time, and it's a good way to get insight into what is going on with evolution.
Oh, great. Thank you so much. We're a little over halfway. How many of you have never been to this kind of competition before? Pretty impressive, huh? So I'm going to tell you a student last year that did his work on mental health in Gilded Age, New Mexico, said that he's used his LoboBites presentation as a springboard to bring his work to more diverse audiences. I've been letting my history scholarship inform my studying with medical professions on North Campus, and I presented at an interdisciplinary conference in Finland over the summer. LoboBites helped cement my confidence in talking to outsiders about my work. Our next speaker is KARISHMA BANSAL. She'll be speaking on the dance of two supermassive binary black holes.
Hi everyone. So what is a black hole? A black hole is a really compact source which has very strong gravitational field. Even the fastest moving object in the universe, the light, cannot escape it. So just to give you an idea what a black hole is, think of our planet, entire Earth, and shrink it to a size of a cherry. Now a cherry with all that mass of the Earth is really compact, really massive and dense. And if you're close to it, you're going to experience very strong gravitational force. So that's a black hole. Now there are two types of black holes in this universe. One is the low mass black hole and the other is supermassive black hole. So the low mass black hole, they have mass of about 10 to 100 times mass of the sun. And the supermassive black hole, they have mass of about few million to billion times mass of the sun. And the mass of the sun is 300,000 times the entire Earth, and that includes you, your house, your property, everything, whatever you want. And that you multiply with million or billion, and that will give you a mass of a supermassive black hole. So you get an idea how massive they are. Now most of the galaxies in the universe, they have a supermassive black hole at their center, including our own galaxy, the Milky Way. And do you know what's going to happen to the Milky Way in another billion years? It's going to collide with its neighbor galaxy, the Andromeda, which also has a supermassive black hole. So what will happen when these two galaxies collide? And so they are going to come together, and the black holes at their center are also going to come together. They're going to form a binary system. Now, I believe you won't be there, right? So you might wonder what's it going to look like. So this is what it's going to look like. So the picture here shows you two black holes in a galaxy. And this galaxy formed as a process of two galaxies merging in the past. And if we study the orbital motion of these two black holes, you can get the combined mass and the period of the system. So that's what we did. We studied the source for 12 years, and we found that these two black holes are orbiting around each other with a period of 28,000 years and with a combined mass of 15 billion solar mass. Now, what's interesting about these sources? So the interesting part about these sources, despite the universe being so big, we should be seeing more and more of such systems, right? But we don't. This happens to be the only most compact supermassive binary black hole ever so far. So we have studied this, and another thing which tells us is how is our universe evolving, what's happening, like our galaxy is not merging, or what's going on. And another interesting aspect is this is the first time we studied the orbital motion of this source. So this is why it was covered in around all around the globe for in 100 news outlets. And another thing is this galaxy is about 2.2 billion light years away from us. A light year is the distance traveled by light in 1 year. And we don't go there, we just stay on Earth, and we just enjoy the show. So thank you.
Thank you, Karishma. Where are you from?
I'm from India.
And was this something that you always knew you'd be interested in?
Well, I always wanted to do astronomy, but I didn't know if I would be working on black holes. And, and actually, like, it's pretty funny. Like, my advisor just gave me data, and then I'm like, oh, what is this? It's like, there are black holes. I'm like, cool, I'll work on them.
Okay. And coming from India, is there something that surprised you about Albuquerque? Is it what you expected?
No, I thought there would be more people, but okay.
Have you adjusted to that?
Oh, yeah, it's been a while now.
Okay.
But I just did too badly, actually, now.
Okay, thank you so much.
Thank you.
Our next speaker is SARINA Baldoni, who will be speaking on cryptocurrencies, myth versus reality.
Thanks. Cryptocurrencies have been described as fake news and highly speculative. And they've also been described as the harbinger of decentralization. But what are some truths about cryptocurrencies? And do they even have value? So let's start with blockchain technology. Blockchain technology is a small but important part of cryptocurrencies. However, it's not only used in cryptocurrencies. Companies such as Google and governmental institutions such as the US Department of Homeland Security also either use blockchain technology or have plans to implement it in the future. What about the size of cryptocurrency? So the market cap of cryptocurrency is $219 billion. And to give you sort of a sense of relativity, that's more than 2 times the size of New Mexico's GDP. So let's move on to some data. This top graph here is called an event study. And we took the top 10 largest cryptocurrencies and tested to see how they reacted to an event here at time 0. Right, that V in the middle. Now, as you can see, there is some trending going on. But there are differences in how the cryptocurrencies reacted. And so these cryptocurrencies having differences is called being heterogeneous. So why does being heterogeneous matter? Well, I'll give you a better visualization here. This graph here is called a correlation. It's a little hard to see, but this is testing how cryptocurrencies move relative to each other. So if it's green, it's positively correlated, which means that they move together. If it's red, it's negatively correlated, or they move oppositely.
And if it's white, it means that they're fairly stable or they don't move much at all. And as you can see, it's not all red and not all green, so there's differences in mix. And we can call this pooling. We can kind of move these into pools of greens and reds, etc. And pools are very important.
Financial modeling has been used for years to describe pools in the stock market. Now, people invest in the stock market because they believe it has value. So if we can use the same methodologies to describe pools in cryptocurrencies, then shouldn't we as investors also believe that cryptocurrencies have value? So there's still sort of a mythological cloud surrounding cryptocurrencies, but there are also facts and research to support it. So I hope that I was able to give you kind of a glimpse into the realities of cryptocurrencies and the potential value that they have. Thank you.
Thanks, SARINA. What got you interested in cryptocurrency?
I'd always heard about it, and I didn't know much about it, and I met a friend a while ago who started telling me more about it, and then I talked to my advisor, and I told him it's something I don't know much about, but I'm interested in, and he said, no way, I'm interested in it too, so we dove right in.
So do you think 5-10 years from now, can you make a prediction? Will cryptocurrency be a common term?
I don't know. I would like to think so. Maybe 20 years.
Okay.
Yeah.
All right. Great. Thank you so much.
Thank you.
Our next speaker is ADELINE FANNI, a novel tool to diagnose and treat Alzheimer's disease.
We all want to live long enough to see our grandchildren and grand-grandchildren grow. When we will reach the age of 65 and older, 10% of us will have Alzheimer's disease, and we might not be able to recognize our own family. To this day, there is no cure, no treatment for Alzheimer's disease, and this leads to a cost of $277 billion here in the United States, just this year, 2018. Yes, you heard it right, billions dollars. But what if I told you there is hope, that we can develop a tool ensuring early diagnosis and early treatment? For the development of such tool, we need to understand what Alzheimer's disease is and which key elements are involved. Alzheimer's disease is a progressive disorder characterized by neuronal cell death. In addition to this cell death, there is a biological marker observed in patients with Alzheimer's disease, which are called protein aggregates. But what does that mean? What are these aggregates? So, okay, we all produce protein in our brain. One of these proteins is called amyloid beta. Patients with Alzheimer's disease will produce more and more amyloid beta, which will accumulate in the brain and then stick to each other to form big clumps. These clumps are called aggregates. During the progression of the disease, these aggregates appear during the early stage. And as you can see on this schematic, they surround the neuronal cells. And they were actually shown to be toxic, to cause cell death. During the progression of the disorder, you can see they grow to make bigger structure, which are called amyloid plaques. So based on this information, you can see how interesting are the small aggregates, the ones who appear early in the disease, 10 to 20 years before we can actually diagnose Alzheimer's disease with our current technology. So we propose to use these small aggregates for early diagnosis. In our lab, we have developed a new molecule called OPE. OPE can actually interact with this aggregate. After interaction, the OPE will fluoresce, they will glow in your brain, and this will help us see the aggregate. The same molecule, the OPE, presents a second characteristic, really, really interesting. After interacting with the aggregate, it will damage them. It will break them down into smaller species, which are believed to be less toxic, and our brain could actually clear them up. So you can see that today there is hope that such technology ensuring early diagnosis and early treatment will ensure that millions of people, such as you and I, will get the chance to see our grandchildren and grandchildren grow. Thank you.
Thank you, Adeline. Tell us where you're from.
So I'm from France.
And what brought you to New Mexico?
So I was always interested by developing new medical tools. I was earlier working on developing antibodies and vaccines, and I was looking for a new challenge, and I feel like Alzheimer's disease is a good one, so I'm here.
Great. Well, I'm going to ask you the same question I asked our friend from India. What surprised you about Albuquerque?
I think the diversity here really impressed me. I wasn't expecting that, and I really love it.
Good. Great. Thank you so much.
Thank you.
Our next speaker is ANDRESSA MAIA, Rethinking Memory, Violence, and Environment in Literature.
Bolsonaro was elected the president of Brazil 2 weeks ago. He is internationally known by his hate speech based in homophobia, racism, and sexism. He is also part of this conservative, populist right wave that we now have in the world, but he's not going to stop my blooming. In 2016, he was one of the senators in Brazilian Congress that voted in favor of Dilma Rousseff's impeachment, and Dilma was the president of Brazil at that moment. And he dedicated his vote to Ustra, an army commander that tortured Dilma during dictatorship while she was fighting against a repressive government. And he's not going to oppress women's blooming. In 2 weeks, he has said twice that he's going to pacify Brazil, just like Duque de Caxias did. And guess what? Duque de Caxias is another army commander that, in the first half of the 19th century, went to the north of Brazil to pacify popular rebellions, killing 20% of North's population, made of indigenous, slaves, and ex-slaves. But in the end, what is the relation between Bolsonaro, Dilma, Ustra, Duque de Caxias, dictatorship, and literature? Well, it is through literature that we can access these silence discourses that has been repressed by the institutionalized truth promoted by governments. Literature helps us to navigate through a sea of remembrances that can materialize not only discourses, but also bodies that has never been found, opening space to mourning and resistance. Marcelo Rubens Paiva resisted when he wrote his book, I Am Still Here, materializing the story of his family. His father was tortured and killed during the dictatorship, and he also fights against the national amnesia that Brazilians still have about the damages that we can still see in Brazilian society nowadays. Luis Rufato also resisted when he wrote his book, Unremembering Me, materializing the letters that his brother sent to his mother while he was living away, being a piece of the dictatorship machinery that was destroying nature, trying to convince Brazilians that the economic expansion that the country was having was something good for everybody, which is not true. Therefore, it is through and at literature that we can use the word resistance to subvert pain into force and courage to fight against repressive governments. We women resist, Brazilians resist, and literature resists. Thank you.
Thank you, Andressa.
Thank you.
Tell us what brought you from Brazil to New Mexico.
Well, you don't need to know from where I am after this speech.
Yeah.
Well, actually, I am here because I discovered that UNM has a great Portuguese program, and they paid my scholarship, tuition, everything. So I'm here.
And what do you think about Albuquerque?
A year ago, I didn't like it. I didn't like green chili. I didn't like the dry weather. I still don't like the dry weather, but now I do like green chili, and I like to live here. I do like to live here, and I was enchanted by the enchanted land, definitely.
Great. And what's next for you?
I don't know. I am kind of working on my PhD applications for now, so I don't know. Let's see what the future is bringing me.
Great. Thank you so much.
Thank you very much.
Our final presentation is Emma Garcia speaking on an engineer's take to the bone ligament interface utilizing novel technology to improve clinical outcomes.
Hi everyone, thanks for sticking around. So when I was a kid, bright-eyed, more imagination than sense, knee-high to a cricket, I decided that I too could fly like the witches and wizards of Harry Potter, and to prove it, I jumped off the back of my dad's truck with the broomstick between my legs and was going to fly off into the sunset. Only I didn't so much fly as go splat on the concrete and ended up snapping a ligament in my ankle. Now this injury still plagues me to this day because despite how common ligament injuries are in the United States, there are no real good methods to repairing them. So older and debatably just as unwise as my broomstick days, I joined a lab here on campus that's looking to create a tissue engineering scaffold to regrow ligament as a better alternative to these repair options. Now, these scaffolds are very similar to the ones you see on the front of buildings during construction, or like the one Michelangelo would have used to paint the ceiling of the Sistine Chapel. They're there to support the workers as they construct the new edifice, and in time are taken away to let the building stand on its own. For the body, however, it's not quite as easy as throwing up some 2x4s or finding a talented Italian painter. Whatever material we use needs to A, not kill the patient, because they tend to appreciate that, and B, mimic whatever tissue we're trying to regrow both mechanically and biologically. Now for ligament, it's even more tricky because the tissue is what we call functionally graded. That means that as it connects to bones, it goes from being stiff or bone-like to viscoelastic, able to stretch in the middle, back to being stiff and bone-like again. Scaffolds traditionally targeting this tissue have been made of one material, but we believe you need a combination of materials and a combination of techniques to capture this complex tissue. So we built what we call our E-spin printer. It's pictured here behind me. It has two tool heads and a movable bed that transitions between them. The two techniques we use with this are called 3D bioprinting and near-field electrospinning. Now 3D bioprinting is very similar to the printers you have in your office, except that our ink is biocompatible and it can be deposited layer by layer to build up a three-dimensional structure. Near-field electrospinning uses an electric field to pull fibers out of solution. So if you think of a cotton candy making machine, you're pulling long strands of sugar out of the syrup in the middle to create kind of a woven mat of syrupy goodness. It's very similar to that, except not edible. So we print and spin materials that are called polymers, which is just a fancy name for plastics. And we characterize how do they behave mechanically. Are they bone-like? Are they ligament-like? And then we see how cells respond to them. Will they attach and begin to regrow the ligament tissue we're looking for? We hope to create a composite scaffold that has two rigid bone-like structures with fibers integrated in between them as a better alternative to a ligament repair option. And we aim by using these engineering techniques and tools to offer new tools for clinicians to use whenever you come into the doctor's office, and ultimately improve patient mobility and quality of life.
Emma, thank you so much. As our last presenter, would you mind saying, what this experience did for you?
It was a really good way to think about how I talk about my research, because everybody knows what ligament is and how much it hurts when you sprain your ankle. But when I would use the term scaffold, I would get a lot of blank looks a lot of times. So it was really good to kind of dial it back and use like layman's terms to reach a broader audience, like my parents.
Great. Yeah, perfect. And what will you be doing next?
I actually just defended my master's yesterday, so I'm graduating real soon.
Oh, all right, the judges have done their thing. We need the audience to do their thing. So you've got perhaps a program. If not, Christine has some. So Christine and Sue, raise your hands so they know who they're looking for. And while Christine and Sue raise their hand, let me give them a big shout out, because I can't tell you how helpful they are to making this all go, as well as Namrata Nepal over here. So thank you. Bill Gannon doing the timer. The graduate studies staff really makes this possible, so I'm very thankful to them. So if I can get your attention, actually, I'll tell you a little bit about the 3-minute thesis, and then I will introduce you to the judges, so it quiets down a little bit. So a 3-minute thesis, a lot of people have probably thought about this idea. How do I convey research in a short time frame? But it got attributed to the dean of the University of Queensland in Australia, and they have some weather sort of similar to ours, and they had a mandated 3-minute shower. And the graduate dean at University of Queensland, during that mandated 3-minute shower, turned over his egg timer and realized, wow, you really can accomplish a lot in 3 minutes if you put your mind to it. And that's where the 3-minute thesis was born at University of Queensland, due to a drought time and a 3-minute mandated shower. So another way to think about your work. I'd like to introduce our judges. So thankful to community members that will come and help us with this. So first of all, on my right, your left, and if you all would turn around and look at the audience, we have Dora Dominguez, who is Senior Economic Developer with the City of Albuquerque. She is also former director of the State of New Mexico's Office of Business Advocacy. And next we have Jim Novak, who's Senior Manager of Cyber Intelligence Research at Sandia National Labs. Jim serves on UNM's alumni board and has an MBA from UNM. And Bobby Baca, who is retired from the U.S. Forest Service and former First Lady of Albuquerque. Her husband, Jim Baca, was mayor in 1997 to 2001. And then finally, we have Representative Mo Maestas, who won again yesterday and is representing District 16 in Bernalillo County. He's served since 2007, and he has a law degree from UNM. So please, another round for these judges. Really appreciate your time. So while they're counting votes, let me also note that the 3-minute thesis is a way to quickly convey the value of your graduate education and research. We need to better communicate the value of our work to the public, and additionally, we need to work together to solve big problems. So currently, the UNM Grand Challenges Initiative, which is sponsored by our Vice President for Research, is an opportunity to broaden our student and faculty perspectives and better serve New Mexico by providing opportunities to collaborate through interdisciplinary research and problem solving. Further, being able to talk about your work to non-experts opens up interdisciplinary opportunities to solve grand challenges. So I hope the 11 of you go off and solve grand challenges. We are looking forward to seeing what you do next. So President Stokes, do you want to go ahead and come up? So we have the judges' scores tallied. We don't have the People's Choice yet. So I thank you very much to our President Garnet Stokes to be here to present these awards. Having the President and the Provost here just shows the importance of this event. So thank you both for being here. So third place winner is Lauren BANSBACH. There's also scholarship money tied with these, so we'll make sure that gets processed. Second place winner is Ty Martin. And our first place winner is Emma Garcia. Now the President and I will do a dance. Well, no. And the People's Choice Award goes to AYUSH SHAHI. Oh, they want one more picture. Can you? For you, Isabel. Thank you very much. And one more thank you to Alumni Affairs for letting us use this beautiful Hodgin Hall facility. We have a beer and wine reception following. We hope you'll stay. There are probably still a few posters up. We'd love for you to stick around for a while, talk to each other about your work, and let's keep this going. Thank you so much.
It's been a really exciting opportunity to present here at the LoboBite competition. It really allowed me to think about my thesis in an accessible, simple way. So I really love forests, I love plants, and I love wild places, and I want to conserve them.
Well, I think this contest is a really great opportunity to figure out a way to take this complicated, in-depth research that we're all doing for grad school and sort of convey that to a general audience, and they can understand sort of, oh, that's what they're really doing at the university, So the biggest thing I feel like is trying to make it something that the audience can relate to, right? So I try to use several different examples of things that relate to common knowledge, like magnets attracting, things like that. So I feel like anytime you can make examples in your talk that the audience might get a better idea of what you're trying to say. I think it's really great. It's a good way for me to figure out what the other students are doing at the university, so like supermassive black holes and things like that. I don't really see much of that very often, so it's cool to see what other students are doing.
Oh, that's a hard question. There were so many good talks. I think I just really tried to find a way to make what I do understandable by a lot of people, because we use a lot of technical terms in my field, so I had to really scale it back and think, what's the easiest way to explain this? I think it's really great. I learned a lot today just in, 20 minutes? And I think it's a really good way for us. We often get caught up in our research to just slow down and think, what am I really doing? I really have always loved math and science, and then I wanted to do something that would have an impact beyond me. And like I said, I have a personal connection to it, so I wanted to find a way to help others in the same situation.